Now, to date, we have learned that you can define life by the various characteristics that it exhibits. Now, one of the major characteristics of life is that life is organized. Now, last time we reviewed extensively the formation of molecules, structural molecules like proteins, carbohydrates, lipids, and fats. All of these molecules come together, in essence, to contribute to the structure of life. Now, today what we're going to be doing is looking at the cell. And the cell is the basic unit of life. Now, the cell was not discovered until the microscope came in. Now, the microscope obviously allowed magnification, and as a result, small things could be seen. When we look at a cell, we have to incorporate the cell theory. Historically, it was Robert Hooke and Leeuwenhoek several hundred years ago, using a very primitive microscopes, that essentially discovered the cell. However, they did not understand the real ramification of their observation. It took several hundred years later for Schleiden and Schwann to come up with the cell theory. In essence, the cell theory says that the basic unit of all of life is the cell, and that one cell results in another cell. And in essence, all cells come from previous cells. You have to understand that every cell has all of the characteristics of life. Now, to get an appreciation for the cell, we can see that cells are specialized and vary in appearance greatly. We're looking, for example, at a yeast cell, not under a regular microscope, but under an electron microscope, and you can see that this cell is actually budding into small cells. Now we're looking at a cross-section of a leaf. A leaf is an organ that carries out photosynthesis. We can see layers of cells of different types. Each is formed into layers of tissue, and each tissue has a given purpose and a given function. Now we're looking at blood cells. You can see the round donut shape are red blood cells, the fuzzy looking cells are white blood cells, and the background cells are actually epithelial cells within the blood vessel. Now you can see all of these cells are different in shape and size, and of course they differ greatly in their purpose for existence. In this slide we see a nerve cell. Notice how peculiar this cell is. It has cilia, which are designed for smell, and the shape is quite different from other cells that we've seen. We're looking at the head of a pin. Notice that on the point of a pin, this is where we want to be, on the point of a pin, we see lots of little aggregates. These little aggregates are actually bacteria, and you can see how small these bacteria are, in the sense that when you see the pinpoint, you cannot see these bacteria. So obviously these bacteria are extremely small, and it takes a microscope to see them. Now, let's get an appreciation for size in life. To start with, we're looking at a scale that shows you humans in the meter range. An egg is about 100th the size of a human. And just to get a better appreciation for size and cell structure and so on, let's pretend that we're looking at the edge of a dime. The edge of a dime is 1 millimeter, or 1,000th of a meter. On the edge of a dime, we are able to place as many as 100 cells. In addition, on the edge of a dime, we could put back to back 1,000 bacteria. On the edge of a dime, we could actually line up 10 to 100,000 viruses, depending on the size of the virus. So you see, life really starts being very, very small. Now, being small is good. Why is that? Why is small good? Because when you look at the size of any object, the surface to volume ratio increases as the object gets smaller. In this sense, a cell is very efficient in getting material in, which is food and oxygen and water and so on, and getting material out, which would be, of course, waste products. So being small is good. Now, to understand the cell, we have to divide cells into two categories: prokaryotic cells and eukaryotic cells. Now, prokaryotic cells evolved about 3.8 billion years ago. It took 2 billion years before the eukaryotic cell evolved. Now, prokaryotic cells make up predominantly bacteria, whereas eukaryotic cells make up most of the visible organisms that we are familiar with. Now, let's compare these two first of all. The DNA in a prokaryotic cell is found in a nucleoid, not surrounded by a membrane, where in a eukaryotic cell, the DNA is found within a confined membrane structure. As far as size, the prokaryotic cell is very small compared to the eukaryotic cell. As far as organization, the prokaryotic cell does not organize itself, where the eukaryotic cell, as you can see to your right, organizes itself into tissue and eventually organs and multicellular organs. As far as metabolism, the two also will differ. And the prokaryotic cell will not have an abundance of membrane-bound organelle, whereas the eukaryotic cell, as you will see in a moment, is full of substructures that are surrounded by membranes carrying out important vital functions. Let's look at the anatomy of the eukaryotic cell. And let's start, first of all, starting from the left, going to the right. If you look at the cell, you'll see that in the center of the cell, we have a nucleus. The nucleus, of course, will have DNA and various other important macromolecules. Coming outside of the nucleus, we see an abundance of substructure, and we'll talk more about the membrane-based substructure in a moment. These membrane-based substructures, or organelle, are floating within a fluid called cytosol, and immersed within the cytosol is a network of fibers and tubules called cytoskeleton, and surrounding everything is a plasma membrane, basically holding the cell together. Now, looking at the anatomy of an animal cell in more detail, let's start from the center and go out. And to start with, we'll look at the nucleus. We see that the nucleus is essentially the control center of a cell. As we come further out from the nucleus, we see a series of membranes with little dots inside. These little dots are ribosomes, and ribosomes are basically protein factories. As we've come further out, we see some other structures called smooth endoplasmic reticulum, and we'll talk about that. We'll talk about these structures in a moment. And then we see some other organelle called mitochondria, the yellow round structures. You'll see the green little structures called lysosomes. You'll see endoplasmic reticulum. You'll see, besides the endoplasmic reticulum, you'll see the Golgi apparatus. So, looking at the anatomy of a cell, we see that it is quite complex to say the least. Now, let's look at a cell as a manufacturing plant. A plant that manufactures a variety of important biological substances. One of the most important will be the macromolecule called proteins. Now, as a factory, many of you have worked at factories, we have a control center where decisions are made. We have a manufacturing plant called the endoplasmic reticulum. We have ribosomes, where the proteins are actually made per se. We have a distribution center called the Golgi complex. We have people that come in and clean up when things aren't going so well, called lysosomes. Now, to power this manufacturing plant, we have mitochondria. And to kind of hold things together, we have a cytoskeleton, which keeps things pretty much intact. And to make certain that the factory is secure, that only certain chemicals come in and out, we have the security gate, of course, which is the cell membrane. Since we're dealing with a factory that's making proteins, let's kind of carry ourselves through a process of making proteins within this factory. To start with, we need to understand the central dogma of biology. And the central dogma of biology says that DNA, which if you remember is present in the nucleus, DNA will transcribe its information to RNA, and the RNA will now go and take that information and translate it into a protein. And all of life evolves around protein molecules. So how are protein molecules made in this particular cell? Let's pretend we're making the molecule insulin, which of course all of you are familiar with insulin because it's an important protein made in biological metabolism. Now, looking at the DNA in the nucleus, there is a gene. As you can see, it's slightly open. The gene for insulin gets a signal from the brain to make insulin. The gene opens up, and the gene will now make RNA. It will transcribe the information or transfer the information from the gene to an RNA molecule. The RNA molecule, which has the information for making insulin, will leave the nucleus, go into the cytoplasm, attach itself to a ribosome, and the ribosome now will start reading the information and make protein, make insulin protein. The insulin protein now will be transferred within the rough endoplasmic reticulum. You can see the green material being made inside endoplasmic reticulum. It will be put into a vesicle, a little round membrane structure, and then transferred to the Golgi apparatus. In the Golgi apparatus, the protein now is going to be modified significantly. A fat material may be added to it, whatever, and eventually the material is going to be released through another vesicle outside and released through the membrane, and the insulin will now move and carry out important functions that insulin carries out. So again, the central dogma of biology says DNA goes to RNA, RNA goes to protein, and we're going to talk a lot about proteins later on. Now, there are other organelles in cell functions. There's the smooth endoplasmic reticulum, there's lysosomes, and there's mitochondria that we have not talked much about, so let's just touch on these for a moment. Now, the smooth endoplasmic reticulum is basically involved in lipid synthesis, making of lipids. This, of course, takes place within the cell. Now, lysosomes carry out a number of functions, but one of the important functions that lysosomes carry out is recycling of material. Here we see a mitochondria that has basically outlived its use coming in contact with the digestive enzymes in the lysosome. The enzymes will degrade the worn out organelle, break it apart, and release the material to be recycled again, or perhaps to be released as waste material. The mitochondria is an important structure, and we will deal with this extensively when we deal with energy metabolism. So, but every cell will have many mitochondria. Depending on the nature of the cell will determine how many mitochondria it has. If a cell has to carry out significant energy metabolism, it will have a lot of mitochondria. For example, which do you think would have more mitochondria? A muscle cell or a fat cell? If you answered muscle cell, you're right. Muscle cells have a lot more mitochondria than a fat cell. Which cell do you think has more mitochondria? A sperm cell or a epithelial cell? If you answer sperm cell, you're right, because the sperm has to move and the mitochondria produce the energy for movement. Now, again, later on, we will study extensively how food is processed, energy is taken out through the mitochondria. Now, a cell is not just a flabby material that looks like a little balloon full of jello. The cell has a cytoskeleton, which gives it shape. And also, the cytoskeleton allows the cell to move because you have single-celled organisms like amoeba that move. And the cell also has a cytoskeleton for the purpose of movement of material within the cell. Essentially, the cell has a highway system for movement of material that is being produced, being processed, and so on. Now, in this slide, we're looking at the various complex types of cytoskeletons that you have. You have microfilaments, you have microfibers, you have microtubules, and as you can see, these cells are all stained to reveal this substructure. But important thing for you to understand is that a cell has a scaffolding that gives it structure, form, and as a result, one cell can look quite different from another cell. Now, this is interesting. This is an artistic illustration of the highway that exists within a cell. It's interesting to see that here we have material that has been made that's being a transport vesicle which contains protein is being moved along the highway within a cell. You probably have traffic cops up and down that control the overall process. And I really mean that you have a regulatory process that moves material within the cell itself through these types of highways. Now, let's look at some of these substructures again. We're looking at cilia that's present on a cell, which obviously gives a cell movement. Here we're looking at a sperm that has a long flagella. The flagella is part of the cytoskeleton that gives the sperm mobility. Here this sperm is trying to penetrate an egg. So we have looked at the anatomy of an animal cell. But we also have plant cells. And plant cells are also eukaryotic cells. But plant cells differ from animal cells. If you look at this illustration, there are three basic parts of a plant cell that differ from an animal cell. First of all, the plant cell, unlike the animal cell, will have a cell wall. The cell wall on the plant cell gives a plant the rigidity that a plant has. And of course, the membrane on an animal cell gives it flexibility. The plant cell will also have a very large central vacuole for storage of food and other metabolic products. The plant cell will also have, as you can see, these green bodies, chloroplasts. And the chloroplasts are responsible for photosynthesis, which is the process, as you will learn, for taking carbon dioxide and converting carbon dioxide to sugars, which plants do quite readily. Now, notice that a plant cell also has all of the other structures that an animal cell has. A plant cell has smooth and rough endoplasmic reticulum, Golgi apparatus, mitochondria, and so on and so on. Now, when a multicellular organism is formed, you can understand that you have lots of cells in different layers, and information that comes from the various parts of an organism has to be disseminated throughout the cell structures. Now, this is carried out through a process that we will discuss in a far more detail later on, cell communication. Cells need to communicate with each other. Now, there are two ways that cells can communicate. One is through a process called plasmodesmata, and the other one is called cell gap junctions. The gap junctions are present in animal cells. The plasmodesmata are present in plant cells. In this illustration, you can see how the tissue of a plant, individual cells come next to each other and channels are formed through the cell membrane. This is referred to as the plasmodesmata. In animal cells, you don't have a cell wall. Instead, what you have is a membrane, and what you have are basically gap junctions that are channels and connections between cells. In this fashion, what happens is information, which usually are chemicals, can be transferred from one cell to another cell, and the overall tissue can act in basically in unison. The table 4.1, as you see, summarizes the various structures, location, and functions of many structures that we have already discussed, and it would be useful for you to key in on the important ones that we have referred to. So, in summary, we have discussed the structure and function of a cell. We've gotten to understand that a cell has complex processes, but the most important, you need to understand that a cell maintains all of the characteristics of life.